Hello everyone. Are we good? Are we on? We're on. I'm really hoping that the stragglers outside, I don't know if they can hear me, are going to come in and rejoin us for the next session because everybody said that they wanted to talk about pricing and scaling, and that is where we're going next. Feel free to come in also and sit in the front row. I don't bite very hard. So everybody come in and fill the front seats as well, because I noticed a lot of people standing in the back during the last session. Okay. Well, we got to keep the show running, so we're going to get started, and I hope everybody will come in for this. Our next session is a panel around solving for scale and price. And I'm not going to read this whole thing at you, but I think this anecdote is really interesting. So last month, an article in Canadian Business wrote, if every bioreactor in current use by the pharmaceutical industry were put toward manufacturing cell-cultured meat for a full year, it would only yield enough food to cover the meat Americans eat in a single day. So that is 1 day of just American consumption. We haven't even moved into the rest of the world with that capacity. So we clearly have a capacity and scale problem. And this next panel is going to be a lot of different perspectives on that problem. The engineering parts, the pricing parts, several different perspectives in the field, and how do we actually sort of engineer and build towards what we hope is a scaled cellular agriculture future. So I'm going to be joined on stage by Kate Krueger, who is going to be our moderator. Kate is at Helikon Consulting, which is an organization that evaluates cutting-edge biotechnology, and excited to welcome Kate back to New Harvest because she was the former research director of New Harvest. Other folks joining me on stage will be Zachary Cosenza from the University of California, Davis, Dr. Anna Chase from Rubik Therapeutics, Dr. Aletta Schnitzler, I really hope I said your name right, from Turtle Tree Labs, and Dr. Zheng Huang from Biotech. So please give our panelists a big round of applause.
Do we need to scoot down? Yeah, thank you. Welcome to our first panel of the day. So I'm really excited to open the conversation with talking a little bit about pricing and scaling. So as you all know, these are some of the most exciting and crucial questions in the space right now for cellular agriculture. And really with this panel, what we're hoping to do is open the conversation. So you'll see this as a recurring trend throughout the conference, not only with just kind of the nuts and bolts, but also with the overarching themes. So this panel today, we have a bunch of different experts from different elements of cell ag. So myself, I'm Kate Krueger, managing partner of Helikon Consulting. So we focus on tech advisory services for investors and others getting into this space. And we focus on a broad range of technologies across cellular agriculture, frontier biotech, and beyond. So everything from bioprocess to synthetic biology and everything in between, which is really a perspective I like to bring to this panel today. And I think that we really are working on with our panelists who have great backgrounds in a variety of the types of tech that go into bioprocess and scaling for technologies. So I'm joined by Aletta Schnitzler, who's an expert not only in cell therapies, but also in all this technologies associated with animal cell culture for food production, Dr. Anna Chase, who is an expert in cell therapy, Zach Cosenza, who's an expert in computational techniques around this area, and Zheng Huang, who's an expert at ARC in bioprocess engineering. So it's always great to get panelists up here that not only can speak to one aspect, but to many. So I'm really hoping that we can have a conversation that focuses on a lot of these spaces we don't usually talk about that kind of fall between the areas of bioprocess and scaling that transcend disciplines. So with that, I'm going to pass the mic to Aletta, who's going to give a little bit of background on herself, and then we can dive into some questions after intros.
Sure. Hi, everyone. So I am coming from Turtle Tree, which is a startup based in Singapore that aims to use precision fermentation to bring milk bioactives to market. We also are working on cell-based milk products. My background prior to that was primarily in developing bioreactor processes, cell culture media materials for the manufacture of cell therapies. And for the last few of my years at Merck KGaA in Darmstadt, Germany, I headed up their cultured meat innovation program. So it's great to be with you.
Hi, everyone. So I'm Anna Chase, co-founder and COO of Rubik Therapeutics. We're a Boston-based cell therapy company developing targeted cell therapies for solid cancers, which is the next frontier in cell therapy, moving beyond blood cancers, which have really had remarkable cure rates with cell therapy so far. And we are engineering cells to recognize tumors in a more precise manner, and also engineering ways to counteract the tumor barriers. So my background prior to Rubik, I have a background in biochemistry and had a stint as a biotech investor, investing in cell therapies and immuno-oncology. And I was also an entrepreneur in residence with a food tech venture builder, actually at Atlantic Food Labs in Berlin, as one of my stints prior to Rubik.
Hello, my name is Zachary Cosenza. I'm just a grad student. So I'm doing my PhD at the University of California, Davis. We have a cultivated meat consortium at Davis where we have a bunch of like 24 PIs and researchers working on cultivated meat. And I work specifically on nonlinear design of experiments and applications to optimizing cell culture media. So it has to do with using artificial intelligence and Bayesian optimization to iteratively search for optimal experiments to run. So you can quickly optimize a bioprocess, whether it's multi-objective or you have some sort of engineering constraints that you want to meet and you need to learn those constraints. So we do a lot of like sort of artificial intelligence plus wet lab work. So, and my background is as a chemical engineer.
My name is Zheng Huang. I'm with ARC Biotech. We're based in Boston. ARC is business-to-business company with focus on developing both hardware and digital infrastructure to enable cultivated meat community to produce meat as commodity. Before ARC, I spent 20 years in the biotech biopharma space, including 13 years at Sanofi, where I was involved in scale-up, tech transfer, and commercial manufacturing of quite a few biopharmaceuticals. And I would say I battled and bruised in the front line of a commercial manufacturing. And those dreadful Friday night call from our manufacturing focus. So and I enjoy to be here and look forward working with the panel to have some good discussion.
Great, well, so excited to have you all here. So as you can tell by these introductions, we have a very techie group of people on the panel who know a lot about the science of bioprocess and engineering, many from a very different context. So the context of using animal cells, particularly human cells, that are grown in bulk for various different applications. And so with that, I'm gonna put Aletta on the spot and talk a little bit about how the technologies from this biomedical space can be repurposed for the cell-based meat space and what those considerations look like.
Yeah, sure. So having made that transition in that order from biopharma manufacturing to cell-based meat and cell-based food products, there's a lot that we can leverage initially, and that's what most companies are doing now, using what's in place for cell culture, animal cell culture, in order to make biologics, as well as cell therapies. But as we all know, the cost and scales of the food industry are completely different than that of the therapeutic industry. So we do need to think beyond the current technologies. There certainly is a lot of proof points around going up to 20,000, 25,000 liter bioreactors for animal cell culture to make a secreted product. But the question is, how much further can that scale for making a cell-based product. And I think that's something that we want to dive into a little bit more in this panel. There are certainly techniques that can use the computational biology to try to start modeling what that will look like. So I think everyone agrees that it's a great starting point, but we really need to think beyond that. And we might need to go in a different direction. So in cell therapies and biologics manufacturing in recent years, there's been more of a trend to go toward single-use componentry. And there's a lot of advantages to that in terms of being able to, especially in a CDMO kind of situation, have more flexibility, have the process completely closed down, and eases the amount of labor. It reduces the burden on steam and clean place, things like that. But the question is that really scalable? And does it resonate with what we're our objectives here in terms of reducing carbon footprint and using renewable supply chains? Maybe the answer is no. There might be a place for single use in early seed train, but I think beyond that, we probably have to think of other things. And then things like perfusion technology. Perfusion is another great way to concentrate the process, which is what we're going to have to do. Right? In the absence of a vascular system, how are we really going to drive these high concentrations that we're going to need to intensify the process? Perfusion is another possibility there, but there's cost of things like the filters. It uses a lot of water. So can we use this as a starting point, but then bring those kinds of technologies, make them more appropriate for the food space? I think that's the challenge that we're trying to tackle here. And then there's some other older technologies that I'm actually interested in that maybe we can enable too, but again, we can get to that.
Yeah. So this is a very interesting conversation because really what we end up talking about is how can we make what we have work in new places? So as Aletta alludes, we do have some really interesting technologies today that can get us closer, bioreactor types, optimization of a lot of these single-use technologies to reusable technologies, both in a climate-friendly way and also in a way that supports lower costs at higher volumes. And a lot of these challenges, in many respects, mirror concerns that people have around cell-based therapies and animal cell cultures in general. So I'm going to hand the mic over to Anna to talk a little bit about what some of those even existing challenges we have for growing animal cells in any context.
Yeah, so definitely the cell therapy space has realized now for a while that having a $500,000 per dose therapy is not really going to be sustainable and for patients. And so I think the big conversation right now is really on, and certainly for companies like mine, we're transitioning to expanding from blood cancer which is some thousands of patients, to tens of thousands of patients in some of the larger disease areas like solid cancers and even beyond that. And so the question there is, how do we transition from so-called autologous therapies, which are cells derived from the patient, to allogeneic therapies, or so-called off-the-shelf, which are cells that can be compatible with many different patients. And so the big discussion in the field now is, what cells will we be using for those off-the-shelf therapies? How quickly will this transition happen? And of course, wrapped into that is then also a bit of the question, and how do we grow these cells and how do we scale? And I would say, I just returned from a conference actually in May, where one of the pioneers of the field, Carl June, from Penn, one of the original cell therapy founders, was talking about his perspective on how quickly this transition to allo is happening. And I think at the moment, there are a couple of cell types that look good. So there's these so-called natural killer cells that can be allogeneic, and also people are now really liking stem cells. And so I think until we as a field, decide what is going to be the horse that we bet on there. It's going to be a bit, the transition is a bit slower in coming than even I want to accept, which is why also our company's platform is actually cell agnostic. So we just put the genes in and can be agnostic to cell type. But yeah, I would love to dig into more kind of some of it's the same thing as Aletta mentioned. The conversation is a lot about optimizing more small scale cell manufacturing, bioreactors, and then also on the side of manufacturing the genes to go into the cells, and also growing those cells to do that is probably one of the areas where maybe we're approaching some of the scales of that we're talking about for food tech.
Great, thanks, Anna. Yeah, I think that's one of the things that I love so much about the cell therapy space is there are other places where people are trying to grow lots of cells for low cost, granted with very different parameters around what low cost looks like, different parameters about how many cells we're talking, but many questions around cell lines, bioreactors, a lot of these considerations that extend really nicely from one space to another. And with that, I have a question for you, Zheng, around bioreactor type and size. So when we start thinking about those components around the cellular agriculture space, what do we think about there, and how do we manufacture that bioreactor for a larger scale?
Yeah, see, current for the commercial cell culture in biopharma space, the biggest bioreactor is around 250,000 liters. And a lot of people say, can you make it bigger? I mean, that's a commonly asked question. My take on that is that size is primarily limited by the downstream capacity, particularly protein A column. So 2 meter is already no joke, and go even bigger make no sense. So the current bioreactor size can easily go another magnitude, and potentially go another order of magnitude. So I think I have a very good chance we'll hit a scale that go even bigger, start making no sense once you're taking all other factor into consideration. So that's my take.
Great.
I mean, it's not easy, but it's not like a nuclear fusion. It will take another 50 years to get there.
Yeah, definitely. Great. Yeah, so it's really great. We got a lot of really talented people working on kind of different solutions to this problem, as we've heard kind of a couple of what those solutions could look like. I'm going to put Zach on the spot a little bit and talk a little bit about different ways that we can make these processes work better. So Zach is a computation expert who's focusing heavily on design of experiments and other computational techniques to optimize processes around cell growth, media costs, and other variables that people care about a lot when they're thinking about feeding cells and growing them in bulk. So Zach, I'm going to ask you to start kind of with what design of experiments is anyway, why people would use it, and what you're hoping to do to make it even better suited for the cell ag space.
Yeah, sure. I don't know about expert, though. I'm the only one without a PhD here. So what design of experiments is, it's like the boring part of every paper that no one really wants to read, because it's after the cool science is done. You're taking the parameter space of inputs that you have, whether it's the concentration of insulin and glucose, or the amount of salt that's in your media, and you're trying to correlate that with an outcome that you want, usually like cell growth or cost. You want to maximize some and minimize others. So design of experiments is the entire field of trying to efficiently explore that design space, either to understand the design space better, so you can go back to the scientists and say that this is the outcome that we're seeing for all these parameters. It goes up and then it goes down. Or if we're trying to do optimization, these are the best sets of parameters. So what I'm doing is I'm applying those techniques to media design. And of course, because the cell culture media that we tend to see in biotech has 30, 50, 100 components, standard methods that you might use to approach the media design problem are insufficient. You can't necessarily break down a 20-dimensional problem into a grid and then test each component individually. I think I calculated yesterday that's like 61 million experiments, and we can't really do that at scale. And if you have competing objectives then you have to like learn a trade-off curve. It's really difficult, so we apply Bayesian methods, machine learning methods to speed up that process. And one of the really cool things about it is you don't necessarily need to know everything about your bioprocess. You have inputs and outputs, and the more you know about it, the easier it is to use design of experiments or other methods to optimize. But we're really going at the problem with like maximum confusion. Like we don't know what the optimal parameters are going to be. There could be 30 of them. What is the best combination of nutrients, for example? So we take that maximum ignorance approach and hopefully turn that into better media quicker. Yeah.
Very exciting. Yeah, so I'm going to open the floor up to everyone and ask you, what is your favorite technology in the space that you're most excited about, that you think will really move the needle on cell bioprocess for cellular agriculture applications?
Do we have to pick just one?
Okay, more than one is fine.
I'm just going to jump in. So I am excited about potentially airlift bioreactors. I don't know, I'm just throwing that out there. I think that that's a really old technology. There's some really cool pictures of huge airlift bioreactors, I think, that were dismantled. It's not my area of expertise, obviously, but if there are technologies now, for example, encapsulation, that can bring the cells together in very, very close proximity, but protect them from the bubble shear forces in airlift bioreactor, we might be able to combine those two things now and unlock and scale up that technology.
Great. So for those of you that may not know about airlift bioreactors, could you maybe give us a little high level on what that looks like?
Sure. Actually, maybe Zheng is the better person to do that. But rather than using impellers to move the liquid around, it actually uses a bubble column. So yes, but that bubble column is pretty intense in terms of its forces. Those reactors are very big, million, maybe million plus liters large. And obviously the KLAs and the gas exchange is a very, very different environment. In order to prevent those microenvironments from happening and get the efficient gas exchange, you have a pretty intense bubble column that's pushing up and moving the whole cell mass around. But again, animal cells have been excluded from that because they don't have a cell wall. So these are typically used for things like mycelium, like the corn product. But maybe we can unlock it for animal cells now.
Yeah, so I think exactly what Aletta's getting at is just how much technology there is out there that with a couple tweaks could, in theory, make a big difference. So instead of having this bioreactor with, again, stirring at the bottom like we see in all the illustrations, large bubbles like in a fish tank, for instance, that go up through a different column that allow us to get cells that get oxygen through different means could be a big difference. So there's a lot of tech out there. And Zheng, I'm going to hand over the mic to you to talk about that or other technologies that have you really excited.
Yeah, other than the hardware, the bioreactor design, one area of opportunity is the automation and control. So today, the typical biopharma need a large number of very skilled people to be able to keep the run going, just keep the run going. It sounds like very much different from people's concept of a high-tech industry. So there's very good room for improve so that you more approaching the automation level of, say, petroleum industry, where you have very few people managing a very large plant. So it's a big opportunity.
Nice. So Zach, what could that automation look like functionally with computational techniques applied? So say you're able to reduce the variables you need to measure by many-fold. What would that look like to be able to implement some of those control questions? What variables do you think could be affected by the technologies you're developing?
Yeah, so one of the big differences, of course, between biotech and the classical petrochemicals and chemicals industry is our understanding of the physics and the biology of what's going on. I think the more that we develop the process knowledge through some of the work that I do, but also the work that's funded by New Harvest and other organizations, we can start to develop something like model predictive control and sort of typical strategies that are used in the petrochemical industry ported over to the biotech industry. And especially when you're talking about the kind of scale the volume scale that we're talking about, you really have to use some of these tools that are more known to like chemical engineers, for example, computational fluid dynamics, and experimental design, and model predictive control and things like that. So what we're talking about is all part and parcel to the same scale-up process.
Great. Yeah. And Anna, I know you're thinking a lot about how you grow lots of cells as well. So
Yeah.
when you start thinking about that, where does your head go?
Yeah, so I actually just wanted to also respond to some of the things on, where we're excited for cellular agriculture, and I definitely think cell therapy in particular, we've been pushing a lot on automation, right, and also having closed systems, which really cuts down on, needing a clean room, and of course a very high cost, specially trained staff, right? That's a huge cost contributor. I guess what I wanted to comment on, where I think maybe cell therapy is at the moment pushing harder and can inspire, or rather cell ag can inspire cell therapy, is really on driving down the cost of things like growth factors and media, because at the moment and I've spoken to people about this, there doesn't seem to be a huge incentive for the biopharma industry to push down those costs. And so I'm super excited just to, about the advancement made there, because as we move into stem cell-based allogeneic therapies, of course, we need growth factors. And that's also, I think I saw something like can be as much as 30% of the cost. So that's this cross-pollination is happening from both sides.
Yeah, very, very excited about that. So for those of you that are maybe getting into the space, growth factors are the most expensive component required to feed cells. So anyone who gets into this space at all starts asking questions about how do we make growth factors cheaper. Growth factors are proteins. They're also required in cell therapy, but as we know from many components of cell therapy, it's okay to have products in there that costs thousands of dollars or more per unit. So how do we change that equation as we scale? And with that question, I'm really excited to ask Aletta a little bit about another component of the bioprocess question that is also really important as we scale, that factors into some of these questions that Zheng was getting at for automation. When I start thinking about automation, the place my head goes first is of course, removing humans, and then the next place it goes is, wow, removing humans probably reduces microbes, which probably makes things cleaner. So with that, I'm going to hand things over to Aletta to talk a little bit about sterility. So when you think about that.
Yeah, sterility is definitely, when I first got into this space, the thing that kept me up most at night. For the whole, how are we going to keep an animal cell culture sterile at these super large scales? I think it's a little bit more straightforward when you're talking about a beer brewing or a microbial fermentation. Animal cells grow much more slowly than microbes, and so if you have a contamination, your culture can very quickly be taken over. There are also potential for viral contaminations, and those are a little bit harder to detect up front, but that can take down your process. So, obviously the first and foremost is that we have to protect the consumer. And luckily there's a lot of testing that can be done and will be done for these products in order to release lots and ensure that they're safe. But the other thing we need to think about regarding costs is protection of the process. And we don't want to lose these big batches, especially if we're pushing up into hundreds of thousands of kiloliters. If you lose that process, that can be devastating. So we do need to think about other ways to maintain sterility and understand more about how the technologies interface with the clean room and what level of clean room do we really need. So I think that we need some more information there, but this will be done on a risk-based approach to understand how to control not only the environment, but close down the process effectively. Then we should start thinking about maybe other materials of construction. Just Anna and I were talking this morning about stainless steel. And what goes into making a large-scale stainless steel fermentation platform. And there, you have to have like specialty welders come in and do these special welds, polish them, make sure that there's no little crevices, that during your steam in place, clean in place process that microbes can hide out in. But maybe there are other materials of construction that would be more effective at reducing that risk, for example. And another one is, are there things that we can do up front in addition to sterilizing the inoculum or the cell culture media around reducing the risk? Things like direct lytic agents, we're getting more and more research around that. So these are peptides and enzymes that can attack, for example, peptidoglycan, which is a very conserved part of bacteria. It's been shown that these direct lytic agents reduce the likelihood to develop resistance. Maybe there's a part somewhere in the process that these can be used as a preventative measure. So yes, we need to rethink sterility because I don't think it's reasonable to have the entire facility being run, as a class 6 or even more sterile environment. That's not going to be cost effective.
Great, yeah, that's so exciting. I love the idea too of using biology to make a system more sterile in and of itself. So if we can use things like enzymes and harness biology to kill and keep clean what we need to. And with that, I'm going to ask Zheng a little bit about other bioreactor considerations. So ARC, of course, is focused on bioreactors in large part, along with other tech, and love to hear what you think a little bit about bioreactor sterility and bioreactors in general for cell ag.
Yeah, for the sterility, biopharma have been dealing with that for a few decades now. So, and the loss of batch obviously is pretty detrimental, but in grand scheme, the loss cultivated meat batch is no more worse than lost a biopharma batch. In biopharma, we can lose $100 million batch. That's the scale you can talk about. So, we can maintain perfusion run for over 100 days in a commercial environment. So I think that's an engineering problem, and that can be solved with, collective effort. So that's the sterility side. For the bioreactor design, that's in the smaller scale, bench scale, there are so many options, and a lot of technology, and sometime there's a new design come out. But when you get into very large scale, it converges to very few. It's a classic scale-up challenge. And then mostly stir tank and the airlift, Aletta mentioned. And that's the most successful large bioreactor today. That doesn't mean cultivated meat industry will not come up with a new design that not only making cells in a stir tank, but also can potentially grow structured meat, like a thick cut steak, at a very large scale. I think that's possible, and there's a lot more innovation space here, and I'm very excited about that.
That's great. That's really, really great. So what questions should we be asking about bioprocess and scale-up that we are not? So I feel like there's a lot of questions people keep coming back to, but there's a lot of tech and a lot of interesting science that we don't talk about nearly as much. So Anna, when you think about this for cell therapy, what comes to mind?
So I think one of the interesting things that's happening in cell therapy is that instead of kind of just focusing on bulk scaling up, say a particular cell type, like the most common being T cells, right, I think now people are really focused on scaling up and facilitating growth of specific subsets of those cells, which are associated with higher therapeutic efficacy. And so I find that actually really interesting because all the experiments so far in cell therapy have really been done kind of in bulk, which is appropriate because we don't yet know which are going to be the best cell types. But I find that's kind of the angle that we're coming from in cell therapy in my company is we're really optimizing and engineering cells, and also trying to decide which cell subsets are then the best to have a therapeutic effect. So I'm wondering whether maybe kind of that same thinking can be applied, not just kind of on the pure bioprocess side, but also kind of taking the, cell therapy is a bit more maybe in the cell biology phase right now, so for better or worse. But that's something that I was just thinking about preparing for this panel.
Great.
Yeah.
That's really exciting. So I'm going to open the same question up to the rest of the panel. What questions should people be asking in cell ag, for those of you that are currently working in the industry, that we haven't been with regards to bioprocess and scale up?
Well, I think this has been asked before, but reducing the cost of growth factors, primarily through recombinant protein engineering, it seems to me that it's possible to do at scale in order to provide the industry with growth factors. Actually, New Harvest has funded research in this area. It's doable. It's generally scalable. That infrastructure just needs to be in place. And as someone who works on sort of managing the trade-off between the cost of the media that I'm developing and its ability to actually proliferate cells, having the growth factors be a lot less expensive, so we can really focus on maximizing growth and discovering what the effects of each of these components are only on growth. That would be really cool. And I'd also like to throw in a shout out to the people doing techno-economic modeling, because that could also really guide the direction of research early on, identifying the most important factors rather than trying to sort of combine them all and determine which set of costs and proliferation and safety and sterility concerns, all balancing them together. What if we had a whole plan model that could do that? So those are some of the interesting things that I'm looking forward to. And that New Harvest is funded, so good job.
Yeah, so current bioreactor run is kind of fit for purpose, mainly oriented to biopharma. I think for cultivated meat, it warrants its own flavor of fit for purpose, that it's from every aspect, from your media prep, your harvest, your cell culture itself, and all the way to the packaging. So everywhere there's opportunities. And initially, out of just say, oh, there's establishment, we can just use. That's nothing wrong. And especially for your scaling up your process, it's perfectly fine to leverage the existing technology. But in order to produce meat as commodity, that takes bolder step in all aspects. And it takes, I would say, an ecosystem from people making raw materials, cultivated meat company develop their own cell lines, and all the way to the people like ARC develop a hardware and a digital infrastructure, and also investment community and all the audience. Yeah. So.
Yeah, and I think that I'd like the theme of sort of purpose-built, and we probably need to start with the product, right? What are we really trying to make here? So we don't need to make a functional piece of muscle like regenerative medicine does, right? We need to meet a certain nutritional profile, protein profile, organoleptic profile, but those cells don't need to be metabolically active and contracting anymore. So I think we have to come at it also from what does the end product look like, and build back from there and put those purpose-built systems in place for that specific product. Because right now we're trying to do tissue regenerative medicine, regenerative medicine kind of techniques and biologics manufacturing techniques, and sort of cramming them together to see how we can use them. But I think maybe we need to start with what are we actually trying to make and at what scale, and build back from there. And I also like Zheng's sentiment about being able to scale up scaffold and structured products as well. Let's kind of put away all of our preconceived notions and see if we can design this thing from scratch.
I love that. So the optimize for delicious.
Yeah.
So that's hopefully what we're going for, is optimize for delicious rather than optimize for functionality inside a living organism, for instance. So that concludes our portion of the panel with pre-set questions. So we're going to open questions up to the audience. and we use Slido for that process. Kristen, should I hand things over to you for Slido, or I can just dive right in? Just dive right in. Okay, here we go. Okay, so hopefully everyone has been using Slido to ask all sorts of fun questions, which we're very excited about. So let's see, where should we start? Okay, so here's a common one. If we assume cell culture products will be blended with plant-based and/or conventional products, oh man, it just disappeared. It is at the top. Very exciting. Thank you. Okay. Will be blended with plant-based and/or conventional meats. In what ways does that challenge our thinking about scaling up?
That's a very interesting question. So I would say that's mostly post-culture processing. There's many ways you can do that, I assume. But if for this adhesion type cell, you could grow on plant-based microcarriers. So that is like from very beginning, it's already blend. But I would say that's an exception. But most commonly, probably, you do that after cell is already matured.
Okay, great.
I see nothing wrong with hybrid products. I'm just putting that out there. I think that hybrid products would be great. I don't think that necessarily has to be purely cell-based. If the scaffold is bringing some plant-based, nutritious, tasty, good texture, aspects, I think that would be fantastic. And I guess the assumption there is that it would reduce the number of cells or biomass that the animal cell culture would need to bring. So I think that could be a really nice starting point.
Great. So many great questions about recent developments in the field. What do you think about Just's announcement about building a 250,000-liter bioreactor for animal cells? How hard is it to jump from current scales to that scale effectively?
I was going to ask Zheng that same question. So.
Yeah, I mentioned the scale up from today's scale by one order of magnitude, which exactly will get you to 250,000. I don't think that monumentally challenge. So it's very practical because the limitation wasn't the upstream to begin with. So.
Great. Do you want to talk a little bit more about the downstream and what goes into that?
I mean, that's the limitation for the biopharma. For cultivated meat, it's open. It's
great.
Yeah.
Great.
There's no constraint.
Great. Yeah. So when we think about that, currently, what volume of medium is used to produce a kilogram of purely cell-based meat, and what is the current price per liter? So there's a lot of variables going on in here I'm going to start with. So I'm going to break it down for people because we're asking a whole lot. I think that we might need a full lecture and chalk talk for. So before we do that, let's start about asking some questions that go into this question, which is what volume of medium do we need to make a kilogram of product? So if you all were going to ask for some inputs to that, where would you go?
Yeah, so I think that you have to consider, well, first, what is the modality? Are these cells in suspension or are they adherent cells? So that's going to factor in. So how close together are the cells, and how closely can you pack them? So there are some limits in suspension culture around due to viscosity and also nutrient gas exchange to how closely you can pack them. And I think those limits, I'm going to throw this number out there, David will correct me. I think it's maybe around 25% cell mass, so that would be 250 million cells per ml. You might be able to push a little bit beyond that. If you're talking about something that's adherent on a scaffold, then you might be able to push that further, and that's why I'm excited about some other technologies that could drive that down. But you also need to deliver the amount of nutrients, right? The cells are growing, they're going to need nutrients. And so for me, it's not necessarily about the volume of media, although if we want to talk about water conservation, yes, that's part of the equation. But it's more about how efficiently can you deliver the nutrients and, as necessary, sweep away the metabolites. Then you can start talking about, well, how tolerant are the cells to those metabolites? Can they be engineered or adapted to be more tolerant? So yeah, it does open up a lot of questions But there are certainly some limitations that are known that we need to work around and design around. And then the cost of the media, I mean, for fermentation, it's already well below, for microbial, already well below $1 per liter. For animal cell culture, I think well below or below $10 per liter is reasonable for certain cell types. Others are more challenging, and we can drive that down even more. And I totally agree that for the growth factors, it's more of a supply chain issue than a technical limitation around driving down that cost.
Great. Zheng, do you want to take a crack at that as well, talking about what components go in there?
I mean, as Aletta mentioned, there's a different calculation, right? So if it's a suspension or anchored cells. One thing about scale up is that just push for cell density. To a certain level, it's further higher, economically become actually not making a whole lot of sense, right? It's not like publish a journal article who can push to 200, 250 million, but the optimal cell density probably lower than that, maybe even significantly lower than that. If you take into consideration all the raw material cost and the water and moving huge volume of liquid, it's have itself its cost. It's a lot of cost. So it's a modeling problem.
Modeling question.
Comes down to. Yeah.
That's great, because we're going to put Zach on the spot anyway with a question up here that's very popular, which is, when optimizing media, how do you address novel ingredients, like substance X that is hugely beneficial but isn't a canonical media component?
Yeah, so we are currently doing that, and we've done research in that. We'll take standard DMEM or Essential 8 or some of the other more canonical published media lists, and oftentimes we'll just throw together growth factor cocktails or some other components that in the literature have shown to increase cell growth. And one of the cool things about, if you're taking a rigorous look at media optimization is you can look at the interactions that those have with all the other components you already have. So you can do process discovery while you're doing optimization. So what in my most current work, we have a growth factor cocktail, and all of these people have said, oh, these are going to be great, and they're not. These particular ones, they didn't work out. So you can actually take a look and do sort of hypothesis testing while you're doing optimization, and look at the interactions between canonical components, salts, amino acids, with these maybe non-standard components like growth factors or plant extracts, which we're also doing research in. So it's totally doable.
A quick shout out to New Harvest, who funds Zach's open research in this space, which means I can ask Zach all sorts of extremely detailed questions about which cocktail mixes do not work well. And I'm going to put you on the spot there. Are there any particular standouts that you've kind of been surprised by, where you think, oh, this component is not nearly as malleable as people think, or something that's been really a surprise in a good way? Yeah,
so I'll give some components that have worked out well and sort of conform with our view of the literature, and some that have not and don't conform. Certain things like EGF, HGF, PDGF, PEGF, some of these components, either because of the concentration ranges used, are too high or too low, or because of interactions with other components, don't necessarily work out, and they're also very expensive. At least in the system that we're using, they don't have a good effect. And then, what we found consistently is glutamine is a really important component, and is probably at a too low concentration in most sort of cell culture that's used in laboratory for non-cell culture, non-cultivated meat-related products, and also a whole lot of carbon sources. Like glucose, sodium pyruvate are also probably way too low. And we sort of knew that this was going to be the case, right? Because if you're only trying to grow cells, then you'd expect that the carbon sources, some of these more basic components, should be higher. But that's just something that we are automatically discovering by looking at the entire design space together. So, and you can read about it in our open access papers.
Yeah, where do people find your paper?
Sorry?
Where do people find your paper?
It's in biotechnology and bioengineering.
Okay. And what is the title on that one?
What is it?
I totally
It's
put
really
you on the
long.
spot. I'm sure Zach will give us it to
It's
us
really
later.
long. If
My
you,
apologies.
you Google
Okay.
my name, it's somewhere. I don't know.
Zach Cosenza and his PI at UC Davis. So
Yep.
definitely
Block.
Google them. David Block, yes.
Yep.
So you can read all about Zach's innovative research in the space. So think about carbon sources and think about amino acids in addition to growth factors. Very, very
Absolutely,
exciting.
yeah.
Very exciting.
Don't take it for granted what the concentration is.
Okay. Okay. Yeah, and the great thing is those are simpler components. So can ask some of those basic questions before getting into other things. So I think we have time for one last question, which is antibiotics. What is so bad about using antibiotics for sterility purposes? Antibiotics in animal farming can cause AMR zoonotic events, but in cultured meat, that's much less likely.
So I guess I'll jump in. I mean, a couple of things. Obviously, you don't want antibiotics to get into your final product. I mean, that's first and foremost. Again, we have to protect the consumer. We do not want even trace amounts of antibiotics getting into these food products. I don't think it's as big of an issue in a cell culture process in terms of generating potentially antibiotic-resistant bacteria, the way that it might be an issue in conventional food systems. But I think that it's more about protecting the consumer. The other thing is that it can actually hide, it can mask a contamination event that you'd want to capture earlier. So that's another thing. And the assumption is that we're designing for systems that can maintain sterility because of their integrity. And we don't need to use antibiotics, at least not at the large scale. We might need to use some antibiotics up front during the biopsy and the initial cell bank procedures, but at scale, the objective is really to not have to use antibiotics. And I would say, first and foremost, it's to protect the consumers.
Great. Great. That's really good context. So are there any companies and groups that are focused on increasing cell yield instead of building the biggest autonomous bioreactor? Isn't that the elephant in the room? Okay. Don't everyone answer at once.
Every cultivated meat company are working on yield. Yeah, I think that's a majority. I can't imagine anyone not working on that, right?
Yeah. In my eyes, it becomes a little bit definitional, right? If you're working on yield, you're working on cost, right? So.
Absolutely, and same thing goes in precision fermentation, titer. I mean, in biologics, manufacturing for therapies as well, titer is the name of the game. And in cell-based meat products, it's going to be, cell mass that you can generate for any given infrastructure. You want to get as much yield out as possible. Absolutely.
Right. Definitely so important. So companies that are increasing, are there any companies that focus on cell yield? Okay, got that one. A lot of companies are aimed at using gene editing to greatly reduce yield instead of, okay, jumped again. My apologies, Slido is killing me here. A lot of companies are using gene editing to greatly reduce or to eliminate the need of growth factors. What do you think the potential or risk is there?
Probably the biggest risk is the product labeling. Once you use CRISPR, things like that, it's automatic GMO product.
Great.
Yeah, and I would also comment, since it's common in cell therapy, I mean, that's just another additional manufacturing complication then, right? It's an additional reagent that's probably tied up with patents you have to license. And in cell therapy, there were some recent results on possible off-target effects of gene editors, the conventional ones. Which may not be quite as detrimental for the cell ag space, but nevertheless something to watch.
I do think there's a lot of nuance here, though, as well, in terms of labeling. Like, GMO versus bioengineered. There are some more specific definitions around that. It's also different in different geographies, how far back your bioengineering has to go. I mean, if it's footprint free, right? So from what I understand in Europe, it's a little bit more stringent about that labeling. Off-target effects are certainly a concern, but if you are removing a component, for example, in a precise way that's footprint free, I think that should be distinguished from just blindly knocking in with, and you don't even know where the genes are going and disrupting other genes. So I think there's a lot of nuance there, but the tools have gotten to the point where they are quite precise, and the genome-wide sequencing technologies have gotten to the point where we can assess these changes. So I don't think it's something that the food industry should, and they haven't been, frankly, been just completely removing from the table. I think there's a lot of good that we can do with these tools if we use the appropriate testing and monitoring methods that are now more and more available.
Yeah, particularly if you use it to reduce the cost of media, then you're making scale up a lot easier. So it may be a trade-off that's worth it.
Great. So the answer is it's not a one-size-fits-all kind of situation. Zheng, do you have something to add on that one?
Yeah, I mean, for example, if you use a genetic modified cell line to produce growth factor as an ingredient for your cell culture, there's scientifically no chance the gene contaminate your product. So
very interesting.
So that's the kind of one area I think is practical.
And when you're in cell therapy and thinking about genetic modifications, and what kind of considerations do you think about in that space, Anna?
Yeah, I mean, definitely some of the same things that were brought up, right? Can we, how precise is the edit? There's basically a lot of people are now switching to beyond just the normal CRISPR that everyone knows to some base editors, they're called. They can really just change very precisely, like one nucleotide at a time. That's sort of the next wave in the field. Yeah, and I think our company is set up such that we're actually not using gene editing, but a lot of companies are seeing success in the space. Caribou Biosciences recently posted some really promising clinical data just a couple of weeks ago. So I definitely see a lot of things ironed out with more precision, and as Aletta alluded to, being able to monitor the changes.
Great. So we're moving past CRISPR. So new frontier. Here we come. So what do you all think about the suitability of hollow fiber bioreactors for cultured meat production?
So unless the hollow fiber is part of your product, otherwise it will be some acceptance issue, and also how to recover cell after you grow the cell. In terms of growth to record level cell density, I think hollow fiber is monumentally advantageous. But just when you scale to very big scale, there's a lot of engineering challenge.
Great. So scaling is really hard. So one question I'm going to start with Aletta, because this is flagged with her, but then expand to the panel is are we at time? Oh, darn. Okay. So many good questions. I get really excited. Everyone, thank you so much for joining us for the bioprocess panel, and thank you all.
Sorry. I know, I feel bad. I'm like running everyone off the stage. That was amazing. Thank you so much. That was great. That was great. I mean, I think we got a really good walkthrough of some of the challenges, but also some of the possibilities that can get us to scale. We're learning lessons from therapeutics with Anna, Zheng's insights into bioreactors and hardware, Aletta's comprehensive look at sterility and antibiotics and other conversations, and Zach maybe literally giving us the secret formula in his paper to media optimization. That's pretty cool. So I think the thing that I take away from this is that we can feel assured that the conversation is happening, that we're grappling with these problems really well, and that there's a lot of optimism in terms of where the tech is developing and the trajectory that I think we're headed in with scale. So very excited. Give our panelists another big round of applause, please. And now I'm going to let you all go to lunch, but please come back here at 2 p.m. for our next session. I promise you it's going to be just as awesome.